Welcome to The Sleepy Pharmacist. This channel shares calm, factual information about medicines and pharmacology. In this video, you'll hear quiet facts about antidepressants, designed to help you learn gently and fall asleep. You can listen actively or let the information fade into the background as you rest. The serotonin transporter, also known as the serotonin transporter protein, contains 12 transmembrane helices. These helices are arranged in a conserved fold with a pseudo-two-fold axis of symmetry. Within the serotonin transporter, a primary substrate binding site is present. This site is located between transmembrane helices 1, 3, 6, 8, and 10. Both serotonin and antidepressant compounds bind at the same primary binding site. This site is known as the S1 binding site. Transport of serotonin requires the presence of sodium ions and chloride ions. These ions are co-transported alongside serotonin. Sodium binds first to the serotonin transporter. Serotonin binds next. Chloride binds last before the conformational change occurs. The serotonin transporter is encoded by a gene called SLC6A4. This gene is located on human chromosome 17. The SLC6A4 gene contains 14 exons. It spans approximately 40 kilobases of deoxyribonucleic acid. The transporter moves serotonin inward with one sodium ion. This process exchanges serotonin for one potassium ion moving outward. Paroxetine has a binding affinity of approximately 0.1 nanomolar at the serotonin transporter. This places it among the most potent compounds in the selective serotonin reuptake inhibitor class. At minimum effective doses, selective serotonin reuptake inhibitors achieve approximately 80% occupancy. This occupancy occurs at striatal serotonin transporters within the brain. An allosteric binding site is also present on the serotonin transporter. This site lies in the extracellular vestibule about 13 angstroms from the central binding pocket. The S enantiomer of citalopram can bind to this allosteric site. Binding there slows ligand dissociation from the primary binding pocket. Tricyclic antidepressants act as competitive antagonists at histamine receptor 1. They also block muscarinic acetylcholine receptors and alpha-1 adrenergic receptors. Among tricyclic antidepressants, doxepin is the most potent antihistamine. Its histamine receptor 1 binding affinity is approximately 800 times greater than that of diphenhydramine. Tertiary amine tricyclic antidepressants include amitriptyline, clomipramine, and imipramine. These compounds show higher affinity for muscarinic acetylcholine receptors and histamine receptors than secondary amines. Secondary amine tricyclic antidepressants include desipramine and nortriptyline. These compounds are active metabolites of the tertiary amine drugs. Clomipramine is considered the most serotonergic tricyclic antidepressant. It preferentially inhibits serotonin reuptake over norepinephrine reuptake. Monoamine oxidase A preferentially oxidizes serotonin, norepinephrine, and epinephrine. These neurotransmitters are its primary substrates. Monoamine oxidase B preferentially oxidizes phenylethylamine and benzylamine. These compounds are classified as trace amines. Both monoamine oxidase A and monoamine oxidase B can oxidize dopamine. Both enzymes can also oxidize tyramine. In early pharmacology research, monoamine oxidase A was observed to be primarily responsible for striatal dopamine degradation in rodent brain tissue. Monoamine oxidase enzymes are localized on the outer membrane of mitochondria. This is the site where their catalytic activity occurs. These enzymes catalyze oxidative deamination reactions. The products include aldehydes, ammonia, and hydrogen peroxide. Chlorgyline selectively inhibits monoamine oxidase A. It has been used as a research tool to distinguish monoamine oxidase enzyme subtypes. Selegiline selectively inhibits monoamine oxidase B at low doses. At higher doses, this selectivity is lost. Monoamine oxidase A is predominantly localized at nigrostriatal dopaminergic axon terminals. This distribution influences its functional role. Monoamine oxidase B is mainly localized in astrocytes and serotonergic neurons. Its distribution differs from monoamine oxidase A. The enzyme cytochrome P4502D6 metabolizes paroxetine, fluoxetine, fluvoxamine, and venlafaxine. It also metabolizes most tricyclic antidepressants. Cytochrome P4502C19 is the primary metabolizing enzyme for citalopram, escitalopram, and sertraline. Genetic variation in this enzyme affects drug levels. Cytochrome P4502C19 converts amitriptyline to nortriptyline. Nortriptyline is an active metabolite. Vilazodone, levomilnacipran, and trazodone are metabolized primarily by cytochrome P4503A4. This enzyme is located in the liver and the intestinal wall. The serotonin 1A receptor is coupled to inhibitory G proteins. Activation inhibits adenylyl cyclase activity. Activation of the serotonin 1A receptor opens inward rectifying potassium channels. This process hyperpolarizes the neuron. Serotonin 1A receptor activation also inhibits voltage-gated calcium channels. This reduces calcium influx into the cell. Chronic administration of selective serotonin reuptake inhibitors induces desensitization of serotonin 1A autoreceptors. This occurs in the dorsal raphe nucleus over 2 to 3 weeks. Desensitization of these autoreceptors disinhibits serotonin neuron firing. This allows increased serotonin release. The proportion of serotonergic neurons showing autoreceptor desensitization increases over time. It rises from about 40% after 3 days to 60 to 80% after 21 days. Chronic antidepressant administration causes downregulation of beta-adrenergic receptor density. This has been observed in rat brain tissue. Long-term antidepressant exposure increases brain-derived neurotrophic factor messenger ribonucleic acid expression. This occurs in the hippocampus and the prefrontal cortex. Expression of cyclic adenosine monophosphate response element binding protein also increases with chronic antidepressant administration. This effect is observed in rat hippocampus. Brain-derived neurotrophic factor activates TRKB tyrosine kinase receptors. Activation initiates downstream signaling cascades. TRKB receptor activation recruits phosphoinositide 3 kinase and phospholipase C-gamma. These enzymes activate additional intracellular signaling pathways. The cyclic adenosine monophosphate protein kinase ACREB cascade regulates transcription of brain-derived neurotrophic factor. It also regulates other neuroplasticity-related genes. Among selective serotonin reuptake inhibitors, fluvoxamine has the highest affinity for sigma-1 receptors. Its binding affinity is 36 nanomolar. Sigma-1 receptor affinity among selective serotonin reuptake inhibitors begins with fluvoxamine. Sertraline ranks second, followed by fluoxetine. Paroxetine shows minimal sigma-1 receptor binding. Its binding affinity is approximately 1,893 nanomolar. Sigma-1 receptors function as endoplasmic reticulum chaperone proteins. They modulate inositol trisphosphate receptor activity. Activation of sigma-1 receptors potentiates nerve growth factor-induced neurite outgrowth. This effect has been observed in cell culture studies. Imipramine received approval from the Food and Drug Administration on April 16, 1959. It was the first tricyclic antidepressant approved in the United States. Fluoxetine received approval from the Food and Drug Administration on December 29, 1987. It was the first selective serotonin reuptake inhibitor approved in the United States. Fluoxetine was first marketed in Belgium in 1986. This occurred 1 year before approval in the United States. Within its first year on the United States market, Prozac generated approximately $350 million in sales. The market launch took place in January 1988. Sertraline received approval from the Food and Drug Administration on December 30, 1991. It became the second selective serotonin reuptake inhibitor approved in the United States. Developed by Pfizer, sertraline later became the most commonly prescribed antidepressant. This status was reached by the year 2016. Paroxetine received approval from the Food and Drug Administration in 1992. The drug was developed by SmithKline Beecham. Fluvoxamine received approval from the Food and Drug Administration in 1994. It was developed by Solvay Pharmaceuticals. Initial approval for fluvoxamine was limited to obsessive-compulsive disorder. Approval for major depression did not occur at that time. Citalopram became available in Europe beginning in 1989. Approval by the Food and Drug Administration followed in 1998. Citalopram was developed by Lundbeck. The differing approval timelines reflect regulatory differences between European and American markets. Escitalopram received approval from the Food and Drug Administration in 2002. It represents the S enantiomer of citalopram. Escitalopram was developed jointly by Forest Laboratories and Lundbeck. Its introduction reflected a life cycle management strategy using chiral switching. Venlafaxine received approval from the Food and Drug Administration in 1993. It was the first antidepressant classified as a serotonin norepinephrine reuptake inhibitor. Wyeth developed venlafaxine. The drug introduced a dual-action pharmacological profile. An extended-release formulation of venlafaxine received approval on October 20, 1997. This formulation enabled once-daily dosing. Duloxetine received approval from the Food and Drug Administration on August 3, 2004. It was developed by Eli Lilly. Duloxetine later received additional indications for neuropathic pain. These approvals expanded its labeled uses. Bupropion received approval from the Food and Drug Administration in 1985. It was developed by Burroughs Wellcome. Bupropion exhibits a dopamine norepinephrine mechanism distinct from other antidepressants. This profile differs from monoamine reuptake inhibition alone. Phenelzine received approval from the Food and Drug Administration on June 9, 1961. It was developed by Parke-Davis. Phenelzine was among the first monoamine oxidase inhibitors approved. These early agents contributed to the development of the monoamine hypothesis. Tranylcypromine was introduced in the United Kingdom in 1960. Approval by the Food and Drug Administration followed in 1961. Tranylcypromine was developed by Smith, Kline, and French. The drug achieved availability on both sides of the Atlantic. In February 1964, tranylcypromine was temporarily withdrawn. The decision followed reports of deaths from hypertensive crisis. This withdrawal underscored the risk of tyramine interactions. The event influenced dietary restrictions associated with this drug class. Esketamine nasal spray received approval from the Food and Drug Administration on March 5, 2019. It became the first glutamate receptor antagonist antidepressant approved. Esketamine was developed by Janssen Pharmaceuticals. Its approval represented a regulatory shift beyond monoamine-based pharmacology. Eli Lilly lost United States patent protection for Prozac in 2001. Generic fluoxetine entered the market that year. Following generic entry, brand Prozac prescriptions declined by approximately 90% within 1 year. This reflected the impact of patent expiration. The brand name Prozac was created by an external naming company in 1975. It represented an early example of non-chemical pharmaceutical branding. During development, fluoxetine was designated compound LY110140. Scientists David Wong, Ray Fuller, and Brian Malloy identified it in 1972. Bernard Brodie founded and directed the Laboratory of Chemical Pharmacology at the National Heart Institute. He led the laboratory from 1950 to 1970. In 1955, Alfred Pletcher, Park Shore, and Bernard Brodie demonstrated that reserpine depletes brain serotonin. This depletion was associated with tranquilizing effects. Robert Bowman and Sidney Udenfriend developed the spectrophotofluorimeter in Brodie's laboratory. This instrument enabled measurement of biogenic amines. The spectrophotofluorimeter allowed detection of small quantities of drugs within the central nervous system. It became a foundational tool in neuropharmacology. Bernard Brodie received the Albert Lasker Award for Basic Medical Research in 1967. The award recognized his contributions to biochemical pharmacology. Julius Axelrod shared the 1970 Nobel Prize in Physiology or Medicine. He shared the award with Ulf von Euler and Bernard Katz. The 1970 Nobel Prize recognized discoveries related to neurotransmitter storage, release, and inactivation. These findings shaped modern neuropharmacology. In 1957, Axelrod demonstrated that catecholamine neurotransmitters are recaptured by presynaptic nerve endings. This process became known as reuptake. Julius Axelrod identified and named the enzyme catechol O-methyltransferase in 1958. The enzyme participates in catecholamine degradation. Axelrod later coined the term reuptake inhibitors. The phrase described mechanisms regulating neurotransmitter levels. Arvid Carlsson shared the 2000 Nobel Prize in Physiology or Medicine. He shared the award with Eric Kandel and Paul Greengard. The 2000 Nobel Prize recognized discoveries concerning signal transduction in the nervous system. These findings advanced understanding of brain chemistry. In 1957, Carlsson demonstrated that dopamine functions as a neurotransmitter. Before this work, dopamine was considered only a norepinephrine precursor. Carlsson also showed that L-DOPA alleviated reserpine-induced immobility in animal studies. These findings were reported in 1957. Arvid Carlsson completed a fellowship at Bernard Brodie's National Heart Institute laboratory. This fellowship occurred in 1955 and 1956. Roland Kuhn identified antidepressant effects of imipramine at the Münsterlingen Psychiatric Hospital. His observations began in 1956. Kuhn published the first clinical results on imipramine on August 31, 1957. The findings appeared in the Swiss Medical Weekly. Imipramine was developed by the pharmaceutical company Geigy. The company was based in Basel, Switzerland. Geigy chemists synthesized imipramine in 1951. The compound was designated G22355. Imipramine was structurally derived from chlorpromazine. Replacement of the phenothiazine sulfur atom with an ethylene bridge produced a dibenzazepine structure. In 1952, clinical trials conducted at Seaview Hospital on Staten Island observed mood elevation in patients with tuberculosis. These patients had been treated with iproniazid. Physicians at Seaview Hospital reported that some patients were dancing in the halls. This behavior was noted despite the severity of their underlying lung disease. Nathan Klein worked as a psychiatrist at Rockland State Hospital. He first reported antidepressant effects associated with iproniazid in 1957. Nathan Klein later coined the term psychic energizer. The phrase described the mood-elevating properties of iproniazid. Iproniazid was marketed under the name Marsalid. It was released as an antidepressant in 1958. The drug iproniazid was withdrawn from use in 1961. The withdrawal followed reports of hepatotoxicity. Joseph Schildkraut published a landmark paper in the American Journal of Psychiatry in November 1965. The paper was titled The Catecholamine Hypothesis of Affective Disorders. In 1967, Alec Coppin published The Biochemistry of Affective Disorders in the British Journal of Psychiatry. This work proposed what became known as the serotonin hypothesis. Zimeldine was synthesized in 1971. It was developed by Arvid Carlsson's research team at Astra in Sweden. Zimeldine became the first selective serotonin reuptake inhibitor to be marketed. It launched in Europe in March 1982. Fluoxetine was identified by scientists at Eli Lilly on July 24, 1972. The discovery was made by David Wong, Brian Malloy, and Ray Fuller. Zimeldine was withdrawn from the global market in September 1983. The decision followed reported cases of Guillain-Barré syndrome. The risk of developing Guillain-Barré syndrome among Zimeldine users was estimated to be approximately 25 times higher than background incidence. This comparison was made against the general population. Nomifensine functioned as a norepinephrine dopamine reuptake inhibitor. The manufacturer, Hoechst, voluntarily withdrew the drug worldwide in January 1986. The withdrawal of nomifensine occurred due to cases of immune-mediated hemolytic anemia. This condition represents a serious blood disorder. Nomifensine-associated hemolytic anemia involved drug-dependent antibodies. These antibodies triggered complement activation and destruction of red blood cells. On March 20th, 1992, the Food and Drug Administration formally withdrew approval for nomifensine. This action confirmed the earlier voluntary withdrawal. Ameneptine is classified as a dopaminergic tricyclic antidepressant. It acts as both a selective dopamine reuptake inhibitor and a dopamine-releasing agent. In December 2022, the Drug Enforcement Administration placed ameneptine into Schedule 1. This category represents the most restrictive level of controlled substances. By the year 2003, ameneptine had been withdrawn from markets in 49 countries. Contributing factors included hepatotoxicity, abuse liability, and severe acne. Tianeptine was initially hypothesized to act as a serotonin reuptake enhancer. This hypothesis preceded identification of its true mechanism. In 2014, researchers at Columbia University characterized tianeptine as a full mu-opioid receptor agonist. Its reported binding affinity was 383 nanomolar. Tianeptine also acts as a full agonist at delta opioid receptors. Its potency at this receptor is lower than at the mu-opioid receptor. Mirtazapine differs structurally from mianserin by the presence of one additional nitrogen atom. This atom was added to one of the tetracyclic rings. Mirtazapine is described as the 6-ASA analog of mianserin. This structural change resulted in a distinct compound. Unlike mirtazapine, mianserin functions as a norepinephrine reuptake inhibitor. Mirtazapine does not exhibit this reuptake inhibiting activity. Gepirone extended release received approval from the Food and Drug Administration in September 2023. It is marketed under the brand name Exua. Before approval, Gepirone had been rejected by the Food and Drug Administration 3 times. These rejections occurred over a development period exceeding 20 years. Gepirone is the first oral selective serotonin 1A receptor agonist approved for major depressive disorder. This mechanism differs from previously approved agents. Nefazodone has been associated with severe hepatotoxicity. Reported incidence of death or liver transplantation ranged from 1 in 250,000 to 300,000 patient years. In June 2004, Bristol-Myers Squibb discontinued Surzone in the United States. Surzone was the brand name for Nefazodone. Nefazodone was withdrawn from European, Australian, and Canadian markets. These actions occurred prior to discontinuation in the United States. Nefazodone is classified as a serotonin antagonist and reuptake inhibitor. This classification reflects its dual mechanism of action. Trazodone is metabolized by the enzyme cytochrome P4503A4. This metabolism produces an active metabolite known as metachlorophenylpiperazine. Metachlorophenylpiperazine functions as a serotonin receptor agonist. It also acts as a serotonin-releasing agent. During trazodone administration, metachlorophenylpiperazine concentrations reach approximately 10% of parent drug levels. This represents a relatively small proportion. Vortioxetine binds to several serotonin receptor subtypes. These include serotonin 3 receptors, serotonin 1A receptors, serotonin 7 receptors, serotonin 1B receptors, and serotonin 1D receptors. Vortioxetine inhibits the serotonin transporter with a binding affinity of 1.6 nanomolar. This represents high-affinity binding. At specific serotonin receptor sites, vortioxetine acts as an antagonist. These include serotonin 3 receptors, serotonin 1D receptors, and serotonin 7 receptors. Vortioxetine also functions as a partial agonist at serotonin 1B receptors. In addition, it acts as an agonist at serotonin 1A receptors. Moclobemide achieves approximately 80% inhibition of monoamine oxidase A within 2 hours of administration. This reflects rapid enzyme inhibition. Monoamine oxidase A activity returns to baseline within 24 hours after the final dose of moclobemide. This recovery reflects reversible enzyme binding. Unlike irreversible monoamine oxidase inhibitors, moclobemide does not require dietary tyramine restrictions. This applies at daily doses between 300 and 600 milligrams. Acetylcholine nasal spray became the first glutamate receptor antagonist approved for treatment-resistant depression. Approval by the Food and Drug Administration occurred on March 5, 2019. Acetylcholine represents the S enantiomer of ketamine. It shows approximately 3 to 4 times higher affinity for glutamate receptors than the R enantiomer. The combination of dextromethorphan and bupropion received approval from the Food and Drug Administration on August 19, 2022. It is marketed under the name Ovaliti. Within this combination, bupropion inhibits the enzyme cytochrome P4502D6. This inhibition increases the bioavailability of dextromethorphan. Dextromethorphan contributes glutamate receptor antagonism. It also provides sigma-1 receptor agonist activity. The selegiline transdermal patch at 6 mg per 24 hours does not require dietary tyramine restrictions. This represents the lowest available dose. At this dose, the selegiline patch bypasses first-pass metabolism. This preserves gastrointestinal monoamine oxidase A activity. At higher transdermal doses of 9 to 12 milligrams per 24 hours, selegiline requires dietary restrictions. This occurs because selectivity for monoamine oxidase B is lost at these doses. Among tricyclic antidepressants, clomipramine shows the highest affinity for the serotonin transporter, also known as the serotonin reuptake transporter. Its binding affinity is approximately 0.14 nanomolar. Imipramine also binds to the serotonin transporter. Its binding affinity of approximately 1.4 nanomolar is lower than that of clomipramine. Amitriptyline exhibits a serotonin transporter binding affinity of approximately 4.3 nanomolar. This value is lower than both clomipramine and imipramine. Desipramine demonstrates a binding affinity of 7.36 nanomolar at the rate norepinephrine transporter. This reflects high affinity for this transporter. At the human serotonin transporter, Desipramine shows a binding affinity of 163 nanomolar. This value is substantially lower than its norepinephrine transporter affinity. Desipramine displays binding affinity greater than 10,000 nanomolar at the rat dopamine transporter. This indicates minimal interaction with the dopamine transporter. Reboxetine binds to the norepinephrine transporter with an affinity of approximately 1 nanomolar. This represents very high affinity at this site. At the serotonin transporter, reboxetine has a binding affinity of 129 nanomolar. This value is much lower than its affinity at the norepinephrine transporter. Reboxetine exhibits binding affinity greater than 10,000 nanomolar at the dopamine transporter. This reflects minimal dopamine transporter binding. Atomoxetine binds to the human norepinephrine transporter with an affinity of 5 nanomolar. It binds to the serotonin transporter with an affinity of 77 nanomolar. At the dopamine transporter, atomoxetine has a binding affinity of 1,451 nanomolar. This indicates low affinity at this site. Bupropion demonstrates a binding affinity of approximately 1.4 micromolar at the norepinephrine transporter. This interaction occurs within the micromolar range. At the dopamine transporter, bupropion binds with an affinity of approximately 2.8 micromolar. Its binding affinity at the serotonin transporter is approximately 45 micromolar. At therapeutic doses, bupropion occupies only 20 to 25% of dopamine transporters. This finding was observed using positron emission tomography imaging. Desensitization of serotonin 1A autoreceptors can be detected as early as 3 days after initiation of selective serotonin reuptake inhibitors. This has been observed in electrophysiological studies. This autoreceptor desensitization has been implicated in the 2 to 3 week delay commonly seen with antidepressant effects. The latency corresponds with receptor adaptation over time. Trazodone acts as a potent antagonist at serotonin 2A receptors. At doses of 50 to 200 milligrams per day, it occupies 90 to 97% of these receptors. Mirtazapine functions as an antagonist at serotonin 2A receptors, serotonin 2C receptors, and serotonin 3 receptors. These actions block activity across multiple serotonin receptor subtypes. In addition, mirtazapine acts as an antagonist at alpha-2 adrenergic receptors. This interaction contributes to its overall mechanism. Mirtazapine also binds as an antagonist at histamine H1 receptors. This receptor interaction contributes to sedative effects. Through alpha-2 adrenergic blockade at presynaptic autoreceptors, mirtazapine increases norepinephrine release. Blockade at heteroreceptors leads to indirect serotonin release. Doxepin shows extremely high affinity at the histamine H1 receptor. Its binding affinity is approximately 0.17 nanomolar. At low doses of 3 to 6 milligrams, doxepin acts as a highly selective histamine H1 antagonist. At these doses, anticholinergic effects are minimal. Sertraline exhibits a sigma-1 receptor binding affinity of 57 nanomolar. This represents moderate receptor affinity. Fluoxetine binds to sigma-1 receptors with an affinity of 120 nanomolar. This value is lower than that observed for sertraline. The cytochrome P4502D6 poor metabolizer phenotype occurs in approximately 7 to 10% of Caucasian populations. This phenotype influences drug metabolism. Among Asian populations, the cytochrome P4502D6 poor metabolizer phenotype occurs in approximately 1%. This frequency is lower than in Caucasian populations. Ultra-rapid cytochrome P4502D6 metabolizer status occurs in 2 to 3% of European populations. These individuals metabolize certain drugs more quickly. In East African populations, the ultra-rapid metabolizer phenotype occurs in approximately 20 to 29%. This prevalence is substantially higher than in European populations. Paroxetine, fluoxetine, venlafaxine, and vortioxetine undergo extensive metabolism by the enzyme cytochrome P4502D6. Genetic variation alters resulting blood levels. Cytochrome P4502C19 extensively metabolizes citalopram and acetylcholine. This process produces much less potent metabolites. Individuals with cytochrome P4502C19 poor metabolizer status have higher serum antidepressant levels. Reduced enzyme activity accounts for this increase. Fluoxetine and its metabolite norfluoxetine inhibit cytochrome P4503A4. Both compounds also inhibit cytochrome P4502D6. After acute administration, fluoxetine has an elimination half-life of 1 to 3 days. This duration is relatively long. With chronic administration, fluoxetine's elimination half-life increases to 4 to 6 days. Continued use extends drug persistence. Norfluoxetine is the active metabolite of fluoxetine. Its elimination half-life ranges from 4 to 16 days. Paroxetine, sertraline, and fluvoxamine have elimination half-lives between 15 and 26 hours. These durations are shorter than those of fluoxetine. Venlafaxine has a short elimination half-life of 2 to 5 hours. This pharmacokinetic property necessitated the development of extended-release formulations. Norfluoxetine retains serotonin reuptake, inhibiting activity comparable to fluoxetine. Both compounds remain pharmacologically active. Odesmethylvenlafaxine represents the active metabolite of venlafaxine. It also exhibits serotonin norepinephrine reuptake inhibition. Desvenlafaxine is marketed as a separate antidepressant. It is chemically identical to odesmethylvenlafaxine. Desipramine forms through endimethylation of imipramine. This metabolic process produces an active compound. The monoamine hypothesis is now regarded as an oversimplification of antidepressant mechanisms. Current models emphasize neuroplastic and network-level changes. Selective serotonin reuptake inhibitors differ in sigma-1 receptor binding. They also vary in anticholinergic effects and cytochrome P450 inhibition profiles. The precise pathway linking transporter inhibition to mood changes remains incompletely characterized. This uncertainty persists after more than 60 years of research. Monoamine oxidase inhibitors continue to be available. Tranylcypromine and phenelzine remain in use after multiple other agents have failed. Genetic polymorphisms in cytochrome P4502D6 and cytochrome P4502C19 create substantial variability. These differences affect metabolism, blood concentrations, and drug response. In October 2004, the Food and Drug Administration required a boxed warning for all antidepressants. The warning addressed increased risk of suicidal thinking in children and adolescents. The boxed warning became effective in January 2005. It applied broadly to all antidepressant medications. The boxed warning introduced in 2004 was based on a combined analysis of 24 short-term placebo-controlled clinical trials. These trials involved more than 4,400 pediatric subjects. The pooled analysis showed an average suicidality risk of 4% among individuals receiving antidepressant medication. The corresponding risk in placebo groups was 2%. In 2006, the Food and Drug Administration extended the boxed warning. The extension included young adults up to 25 years of age. The warning was expanded again in 2007. It stated that major depressive disorder itself is associated with increased suicide risk. Nefazodone was discontinued in Canada effective November 27, 2003. Hepatotoxicity concerns prompted this action. The Food and Drug Administration required a boxed warning for nefazodone-related hepatotoxicity in 2002. This requirement preceded market withdrawal. On August 24, 2011, the Food and Drug Administration issued a drug safety communication regarding citalopram. The communication stated that doses above 40 milligrams per day should not be used. The dose restriction for citalopram was based on dose-dependent prolongation of the QT interval on electrocardiography. This effect represents a cardiac rhythm concern. On March 27, 2012, the Food and Drug Administration revised the citalopram prescribing label. The maximum recommended dose was reduced to 20 milligrams per day for individuals over 60 years of age. The 20 milligram citalopram maximum also applies to individuals with hepatic impairment. It applies to cytochrome P4-502C19 poor metabolizers and to those taking cytochrome P4-502C19 inhibitors. The risk evaluation and mitigation strategy for esketamine nasal spray was approved by the Food and Drug Administration on March 5, 2019. It was modified on June 25 of the same year. This risk evaluation and mitigation strategy requires healthcare settings to obtain certification. It also requires patient enrollment in a centralized registry. The strategy mandates 2-hour post-dose observation following each esketamine administration. This monitoring occurs after every treatment session. Esketamine carries boxed warnings for sedation and dissociation. Additional warnings address abuse and misuse potential. Esketamine is classified as a Schedule III controlled substance under the Controlled Substances Act. It also carries a warning for suicidal thoughts and behaviors in young adults. The Food and Drug Administration approved fluoxetine for pediatric use in individuals aged 8 to 17 years. This approval occurred between 1997 and 2002. Fluoxetine became the first selective serotonin reuptake inhibitor approved for pediatric major depressive disorder. This approval represented a significant regulatory milestone. On October 15, 2004, the Food and Drug Administration determined that nefazodone had not been withdrawn for reasons of safety or effectiveness. This determination allowed continued generic manufacturing in the United States. Agomelatine acts as an agonist at melatonin receptor 1 and melatonin receptor 2. It also functions as an antagonist at serotonin 2C receptors. Antagonism of serotonin 2C receptors by agomelatine disinhibits dopamine and norepinephrine release. This effect occurs within the prefrontal cortex. Agomelatine displays melatonin receptor agonist affinity of approximately 0.1 nanomolar. Its serotonin 2C antagonist affinity is approximately 631 nanomolar. The European Medicines Agency granted marketing authorization for agomelatine in February 2009. It is marketed under the brand name Valdoxan. European regulatory monitoring requirements for agomelatine include liver function testing at baseline. Additional testing occurs at 3, 6, 12, and 24 weeks after initiation. Agomelatine is contraindicated when liver transaminase levels exceed 3 times the upper limit of normal. The European Medicines Agency initially rejected agomelatine's first marketing application in July 2006. The decision cited insufficient demonstration of efficacy. Novartis discontinued agomelatine development in the United States in October 2011. This decision followed disappointing Phase 3 clinical trial results. Brexanolone received approval from the Food and Drug Administration on March 19, 2019. It was the first drug specifically indicated for postpartum depression. Brexanolone is an intravenous formulation of allopregnanolone. Allopregnanolone is an endogenous neurosteroid that modulates gamma-aminobutyric acid type A receptors. Administration of brexanolone requires a continuous 60-hour intravenous infusion. The infusion uses stepped dosing at 30, 60, and 90 micrograms per kilogram per hour. The 60-hour infusion duration allows titration to manage sedation tolerance. It also reduces the risk of withdrawal symptoms after discontinuation. The risk evaluation and mitigation strategy for brexanolone mandates continuous pulse oximetry during administration. Sedation assessments are required every 2 hours during non-sleep periods. Zuranolone received approval from the Food and Drug Administration on August 4, 2023. It became the first oral medication specifically indicated for postpartum depression. Zuranolone is a synthetic analog of allopregnanolone. It is administered as a once-daily oral dose for 14 days. Adequate absorption of zuranolone requires evening administration with fat-containing food. Opipramole has a dibenzazepine tricyclic chemical structure. Despite this structure, it does not inhibit monoamine reuptake. Opipramole acts primarily as a high-affinity sigma-1 receptor agonist. Its binding affinity ranges from approximately 0.2 to 0.3 nanomolar. In Germany, opipramole is marketed under the brand name Incidon. Rapastanel is an amidated tetrapeptide composed of threonine, proline, and threonine. Rapastanel acts as a positive allosteric modulator of the N-methyl-D aspartate subtype of glutamate receptors. It binds at a site distinct from the glycine co-agonist site. Allergan acquired Rapastanel through its purchase of Naurex in 2015. The acquisition cost was $560 million. On March 6, 2019, Allergan announced that Rapastanel failed to differentiate from placebo. This outcome occurred across 3 phase 3 clinical trials. The compound known as AV101 is converted by kynurenine aminotransferase enzymes in astrocytes. This conversion produces 7-chlorokinurenic acid. 7-chlorokinurenic acid acts as a potent antagonist at the glycine B site of glutamate receptors. A V101 failed a phase 2 randomized trial in treatment-resistant depression. Compass Pathways received breakthrough therapy designation from the Food and Drug Administration in October 2018. This designation was granted for psilocybin in treatment-resistant depression. The Usona Institute received breakthrough therapy designation in November 2019. This designation applied to psilocybin in major depressive disorder. Compass Pathways announced achievement of the primary endpoint in its first phase 3 psilocybin clinical trial. This announcement occurred in January 2025. The Food and Drug Administration granted breakthrough therapy designation for MDMA-assisted psychotherapy on August 16, 2017. The designation applied to post-traumatic stress disorder. On June 4, 2024, a Food and Drug Administration Advisory Committee voted 9-2. The committee concluded that available data did not demonstrate effectiveness for MDMA. Concerns raised by the Food and Drug Administration included functional unblinding and missing safety data. Additional concerns involved abuse potential and ethical violations at a trial site. Indolpine was introduced in France in 1983 under the brand name Upstain. It was withdrawn in 1985 due to agranulocytosis. Indolpine entered the market after zimaldine had already been introduced as a selective serotonin reuptake inhibitor. Alaproclate was developed by Astra AB. Development was discontinued after hepatotoxicity was observed in rodent studies. Alaproclate demonstrated dual pharmacological activity. It acted as both a selective serotonin reuptake inhibitor and a non-competitive glutamate receptor antagonist. Femoxetine was developed by Ferrocen in 1975. Its development was halted in favor of focusing on the structurally related selective serotonin reuptake inhibitor paroxetine. Indoloxazine was marketed in Japan and South Korea from 1988 through 1998. It was reportedly withdrawn due to lack of effectiveness. Indoloxazine acted through multiple mechanisms, including serotonin release and inhibition of norepinephrine reuptake. It also functioned as a glutamate receptor antagonist. Methoxamine was marketed in France, Spain, and Morocco beginning around 1990. It was withdrawn between 1999 and 2000 due to hepatotoxicity. Methoxamine acted preferentially as a dopamine reuptake inhibitor. Its serotonin reuptake inhibition was weaker. Pivigabine was launched in Italy in 1997 under the brand name Tonerg. It is now believed to act through inhibition of corticotropin-releasing factor. Minoprine was marketed in France and was withdrawn in 1996. The withdrawal followed reports of convulsions. Minoprine functioned as a reversible monoamine oxidase A inhibitor. It also displayed weak acetylcholinesterase inhibitory activity. Vicodin acted as a potent serotonin-releasing agent. It also exhibited positive allosteric modulator activity at gamma-aminobutyric acid type A receptors. Vicodin was never marketed. Research focused primarily on its potential use in alcoholism. Litoxetine exhibited selective serotonin reuptake inhibitor activity along with antagonism at serotonin 3 receptors. Its serotonin 3 receptor binding affinity was 85 nanomolar. Development of litoxetine for depression ceased in the late 1990s. Development was later reinitiated for urinary incontinence beginning in 2017. Lubazidone reached Phase 2 clinical trials before being discontinued in 2001. It combined selective serotonin reuptake inhibition with serotonin 2 A receptor antagonism. Lubazidone had a serotonin transporter binding affinity of 21 nanomolar. Its serotonin 2 A receptor binding affinity was 86 nanomolar. Milnacipran received Food and Drug Administration approval in January 2009 for fibromyalgia only. It is not approved for major depressive disorder in the United States, despite approval for depression in France and Japan. Reboxetine received preliminary Food and Drug Administration approval in 1999. This approval was later reversed after additional clinical studies raised concerns about efficacy. A 2010 systematic review found that published reboxetine data substantially overestimated benefit compared with placebo. The degree of overestimation reached up to 115%. Nearly 3/4 of patient trial data for reboxetine remained unpublished. This case represents a notable example of publication bias in pharmaceutical research. Cetiptaline was launched exclusively in Japan in 1989 by Mochida Pharmaceutical. It is marketed under the brand name Tricyclic. Cetiptaline acts as an alpha-2 adrenergic receptor antagonist and a serotonin 2 receptor antagonist. It also functions as a norepinephrine reuptake inhibitor and a histamine H1 receptor antagonist. Aptazapine is approximately 10 times more potent than mianserin at alpha-2 adrenergic receptor antagonism. It reached clinical trials in the 1980s but was never marketed. Eptaperone is a full agonist at serotonin 1A receptors. Its binding affinity at this receptor is 4.8 nanomolar. Eptaperone displays intrinsic activity approximately equivalent to serotonin at the serotonin 1A receptor. No triple reuptake inhibitors have been approved for depression. Failed compounds include NS2359, imidafidine, and DOV216303. Failures of triple reuptake inhibitors have been attributed to lack of superior efficacy over existing treatments. Additional contributing factors include abuse liability related to dopamine, reuptake inhibition, and cardiovascular effects. Tianeptine shows no measurable affinity for the serotonin transporter. The original classification of tianeptine as a serotonin reuptake enhancer was a historical misnomer. The noradrenergic and specific serotonergic antidepressant class includes mirtazapine, mianserin, and cetiptaline. These compounds are tetracyclic piperazine azapine analogs. Marketed serotonin antagonist and reuptake inhibitors beyond trazodone and nefazodone include etoperidone. They also include lorpiprazole and mepiprazole. Prozac Weekly is a 90 mg delayed-release capsule containing enteric-coated granules. When switching to Prozac Weekly, the 90 mg dose is initiated 7 days after the final daily 20 mg fluoxetine dose. Desvenlafaxine received Food and Drug Administration approval in February 2008. It represents a life cycle extension of venlafaxine. Desvenlafaxine is metabolized primarily through glucuronidation by uridine diphosphate glucuronidyltransferase enzymes. This pathway bypasses cytochrome P4502D6 metabolism. Levomilnacipran is the 1s2R enantiomer of racemic milnacipran. It received Food and Drug Administration approval in July 2013. Levomilnacipran is 50 times more potent at inhibiting norepinephrine reuptake than the 1R2S enantiomer. It exhibits approximately 2-to-1 selectivity for norepinephrine over serotonin reuptake inhibition. Vilazodone received Food and Drug Administration approval on January 21, 2011. It is classified as a serotonin partial agonist reuptake inhibitor. Vilazodone bioavailability increases by approximately 64 to 85% when administered with food. Symbyax received Food and Drug Administration approval in 2003 for bipolar depression. It is a fixed-dose combination of the atypical antipsychotic olanzapine and the selective serotonin reuptake inhibitor fluoxetine. Symbyax later received Food and Drug Administration approval in March 2009 for treatment-resistant depression. Gepirone was rejected by the Food and Drug Administration in 2002 and again in 2004. A third rejection occurred in 2007. The Food and Drug Administration cited inadequate evidence of effectiveness for Gepirone. Only 2 of 15 submitted clinical trials were considered positive. Michigan became the first United States state to schedule tianeptine. It was classified as a Schedule II controlled substance effective July 4, 2018. Alabama upgraded tianeptine to Schedule 1 on November 14, 2021. Ohio, Georgia, and Minnesota have classified tianeptine as Schedule 1 substances. Tennessee, Arkansas, and Oklahoma have classified tianeptine as Schedule 2 substances. Nifazodone was voluntarily withdrawn by Bristol Myers Squibb on June 14, 2004. The company cited declining sales rather than safety concerns. Generic nifazodone remains available in the United States following the withdrawal of the branded product. Ammoneptine was placed in Drug Enforcement Administration Schedule 1, effective November 2022. Scheduling was based on high abuse potential and lack of accepted medical use. Sertraline shows the highest dopamine transporter affinity among selective serotonin reuptake inhibitors, with a dopamine transporter binding affinity of approximately 25 nanomolar. Fluoxetine acts as a competitive inhibitor of cytochrome P4502D6, also known as CYP2D6, with a binding affinity of 68 nanomolar. Norfluoxetine also inhibits cytochrome P4502D6 competitively, with a higher binding affinity of approximately 35 nanomolar. Paroxetine causes mechanism-based irreversible autoinhibition of cytochrome P4502D6. At doses between 20 and 30 milligrams, approximately 93% enzyme inactivation occurs. Fluvoxamine is the most potent inhibitor of cytochrome P4501A2, also called CYP1A2, among selective serotonin reuptake inhibitors. Its inhibition constant ranges from 0.12 to 0.24 micromolar. Trimipramine demonstrates negligible inhibition of serotonin, norepinephrine, and dopamine reuptake at therapeutic concentrations. The antidepressant mechanism of trimipramine may involve dopamine 2 receptor blockade. Additional contributions may include alpha-1 adrenergic receptor antagonism and histamine H1 receptor antagonism. Clomipramine undergoes metabolism to desmethylclomipramine. This metabolite displays a more noradrenergic receptor profile than the parent compound. Racemic ketamine remains approved by the Food and Drug Administration only for anesthesia, a status maintained since 1970. No regulatory pathway has been pursued for labeling racemic ketamine for depression. Brexanolone is marketed under the brand name Zulresso. The risk evaluation and mitigation strategy for Zulresso restricts administration to certified healthcare settings. Zuranolone is a synthetic analog of the endogenous neurosteroid allopregnanolone with oral bioavailability. Opipramole exhibits sigma-1 receptor binding affinity that is more than 2,000 times greater than its affinity for monoamine transporters. Rapistinel functions as a positive allosteric modulator of the glutamate receptor, a mechanism that distinguishes it from direct glutamate receptor agonists. The acquisition of Norex by Allergan represented one of the largest investments in a glutamate-targeting antidepressant development program. AV-101 was developed by VistaGen Therapeutics as a prodrug designed to enhance brain delivery. The Phase 3 clinical trial conducted by Compass Pathways for psilocybin in treatment-resistant depression reported success on its primary endpoint after 7 years of development. The Food and Drug Administration Advisory Committee vote on MDMA in June 2024 was not binding on the agency's final regulatory decision. A granulocytosis associated with indalpine led to its withdrawal within 2 years of market introduction. Zimildine reached the market before indalpine, becoming the first selective serotonin reuptake inhibitor to do so. Astra AB discontinued aliproclate based on preclinical hepatotoxicity findings rather than adverse events observed in humans. Pharosan's decision to halt femoxetine development allowed clinical resources to be redirected toward the selective serotonin reuptake inhibitor paroxetine. Indaloxazine remained on the market in Japan and South Korea for approximately 10 years before withdrawal due to concerns about effectiveness. Methoxamine's preferential inhibition of dopamine reuptake distinguished it from many antidepressants available during the same period. Pivagibine's mechanism was later reclassified from gamma-aminobutyric acid-related activity to inhibition of corticotropin-releasing factor. Minoprine was withdrawn from the French market after several years of use due to its convulsive liability. Vicquiline's combined activity as a serotonin-releasing agent and gamma-aminobutyric acid type A receptor modulator was primarily studied in alcohol use disorder. Litoxetine's development trajectory shifted from depression toward urinary incontinence over a period of approximately 2 decades. Lubazidone combined serotonin reuptake inhibition with serotonin 2A receptor antagonism. This pharmacological profile resembled that of trazodone. Milnacipran's regulatory status differs between regions. In Europe, its approved indications include depression. The reversal of reboxetine's preliminary Food and Drug Administration approval followed post-submission reviews of efficacy data. Publication bias in reboxetine trials resulted in most patient data remaining unavailable for independent evaluation. Cetiptaline exhibits 4 distinct receptor activities spanning adrenergic, serotonergic, noradrenergic, and histaminergic systems. Aptazipine's greater potency at alpha-2 adrenergic receptors compared with mianserin did not result in market approval. Eptaperone demonstrates intrinsic activity at serotonin 1A receptors comparable to that of endogenous serotonin. Development programs for triple reuptake inhibitors encountered recurring challenges across multiple candidate compounds. The original pharmacological classification of tianeptine was based on early in vitro findings that were later shown to be inaccurate. The tetracyclic piperazine-azepine structure is shared among all marketed noradrenergic and specific serotonergic antidepressants. Etoperidone, lorpiprazole, and mepiprazole represent serotonin antagonist and reuptake inhibitors beyond the most commonly discussed agents. The enteric coating applied to Prozac weekly granules enables the delayed-release formulation used for once-weekly dosing. Desvenlafaxine's metabolism through glucuronidation represents a pathway distinct from cytochrome P450-mediated oxidation. The potency difference between levomilnacipran and its mirror image enantiomer is approximately 50-fold for norepinephrine reuptake inhibition. Vilazidone's classification as a serotonin partial agonist reuptake inhibitor reflects its dual binding properties. The fixed-dose combination used in Symbyax pairs the atypical antipsychotic olanzapine with the selective serotonin reuptake inhibitor fluoxetine. Gepirone accumulated 3 separate Food and Drug Administration rejections over a 5-year period. State-level scheduling of tianeptine in the United States began in Michigan and later expanded to multiple additional states. The withdrawal of branded nifazodone by Bristol-Myers Squibb did not eliminate the availability of generic formulations in the United States. Ammoneptine was placed in Drug Enforcement Administration Schedule 1 in November 2022. Norfluoxetine exhibits greater inhibitory potency at cytochrome P4502D6 than fluoxetine. Paroxetine's inhibition of cytochrome P4502D6 occurs through an irreversible mechanism rather than competitive inhibition. Fluvoxamine's inhibition constant for cytochrome P4501A2, also known as CYP1A2, places it among the most potent inhibitors of this enzyme. Trimipramines receptor profile lacks the monoamine reuptake inhibition typical of most other tricyclic antidepressants. Metabolism to desmethylclomipramine shifts the pharmacological profile of clomipramine treatment toward noradrenergic activity. Racemic ketamine's indication for anesthesia has remained unchanged since its original approval by the Food and Drug Administration in 1970. Based on available records, the regulatory pathway for racemic ketamine in depression has not been pursued. Brexanolone's 60-hour infusion protocol incorporates both gradual uptitration and tapering phases. Zuranolone's 14-day oral treatment course contrasts with brexanolone's single 60-hour infusion approach. Opipramole demonstrates high sigma-1 receptor affinity alongside minimal activity at monoamine transporters. Rapastanol binds to a site on the glutamate receptor that is distinct from the glycine coagonist binding site. In March 2019, Allergan announced the failure of 3 Phase 3 rapastanol trials simultaneously. 7-chlorokinurenic acid produced from AV101 acts at the glycine B site of glutamate receptors. Breakthrough therapy designation for psilocybin was granted separately to 2 different organizations by the Food and Drug Administration. The Food and Drug Administration Advisory Committee identified 4 distinct concerns related to MDMA. Indolpine remained on the French market for approximately 2 years before withdrawal. Aliproclate's glutamate receptor antagonism was non-competitive in nature. Structural similarities between the selective serotonin reuptake inhibitors, paroxetine and femoxetine influenced Ferrisin's development priorities. Indoloxazine combined serotonergic, noradrenergic, and glutamatergic mechanisms. Methoxamine was marketed across 3 countries in Europe and North Africa. Pivigabine was launched in Italy under the brand name Tonerg. Minoprine inhibited monoamine oxidase A through a reversible mechanism. Vicqualine's research focus on alcoholism limited its development as an antidepressant. Litoxetine serotonin, 3 receptor affinity of 85 nanomolar accompanied its reuptake inhibition activity. Lubazodone's development was discontinued during Phase 2 clinical trials in 2001. Milnacipran holds regulatory approval for depression in both France and Japan. The 2010 Systematic Review of Reboxetine quantified publication bias at up to 115% benefit overestimation. Mochida Pharmaceutical launched C-tiptoline exclusively for the Japanese market. Clinical trials of aptazapine conducted in the 1980s did not result in regulatory approval. Eptiperone's full agonist activity at serotonin 1A receptors distinguishes it from partial agonists. Cardiovascular effects contributed to the failure of triple reuptake inhibitor development programs. Concerns regarding abuse liability of triple reuptake inhibitors were linked specifically to dopamine reuptake inhibition. The noradrenergic and specific serotonergic antidepressant class includes 3 marketed agents. Prozac Weekly utilizes enteric-coated granules contained within a capsule formulation. Desvenlafaxine bypasses cytochrome P4502D6 metabolism through primary glucuronidation. Levomilnacipran exhibits approximately 2-to-1 selectivity for norepinephrine over serotonin reuptake inhibition. Vilazodone absorption increases substantially when administered with food. Symbyax received its treatment-resistant depression indication 6 years after approval for bipolar depression by the Food and Drug Administration. 15 gepirone trials were submitted to the Food and Drug Administration during its development program. 6 United States have enacted scheduling controls for tianeptine. 3 states classify tianeptine as Schedule 1, while 3 states classify it as Schedule 2. Generic nefazodone remained available in the United States following withdrawal of the branded formulation. Amineptine's placement in Schedule 1 cited both abuse potential and lack of accepted medical use. Sertraline exhibits a dopamine transporter binding affinity of approximately 25 nanomolar. Fluoxetine shows a cytochrome P4502D6 binding affinity of 68 nanomolar. Norfluoxetine displays a higher cytochrome P4502D6 binding affinity of 35 nanomolar. Paroxetine administered at 20 to 30 milligrams produces approximately 93% cytochrome P4502D6 inactivation. Fluvoxamine's cytochrome P4501A2 inhibition constant ranges from 0.12 to 0.24 micromolar. Trimipramine may exert antidepressant effects through dopamine 2 receptor blockade. Additional effects of trimipramine may involve alpha-1 adrenergic receptor antagonism. Trimipramine may also exert effects through histamine H1 receptor antagonism. Clomipramine undergoes metabolic conversion to desmethylclomipramine in the body. Desmethylclomipramine displays a more noradrenergic receptor profile than clomipramine itself.